DNA methylation is one of the most studied epigenetic signatures. Many cancers are characterized by changes in DNA methylation levels, which are associated with aberrant gene expression and other genomic abnormalities. Long interspersed nuclear elements are repetitive transposable genomic sequences that normally have high levels of methylation. However, they often become hypermethylated in cancer, which results in their activation and leads to chromosomal instability. In this study, we treated mesenchymal stem cells with Osteosarcoma-Derived extracellular vesicles to see whether cancer EVs can affect LINE-1 methylation in MSCs.
Fetal bovine serum, which is an integral component of mammalian cell culture media, contains a large number of bovine-derived EVs. These EVs will interfere with the analysis of EVs from cells of our interest. Therefore, when growing cells for EV isolation, it is important to use EV-depleted FBS. Take FBS in ultracentrifuge tubes and place them in ultracentrifuge buckets. Balance the buckets to within 10 mg of each other prior to loading them on a swinging rotor. Place the rotor in the ultracentrifuge and run at 100,000 G for 19 hours at 4 degrees Celsius. After the run, carefully collect the light-colored upper layer of the supernatant and transfer it